So it's customary for the speaker to thank the conference organizers, and I want to do that particularly because when I walked into the room, Lindsay shared with me that at the time we were only 2 hours and a half away from cocktails. So that was something to think about before we get started here. Seriously, I'd also like to thank all of you for taking a few minutes with us today to listen to the StemBioSys story. This is one I love to tell, and I hope you'll enjoy hearing it. I want to start with what I'm calling here a reintroduction to StemBioSys. For many of you, if you've attended this conference before, you may have heard some of this. For those of you who have not heard this before, I'll try and keep this, give you enough here to give you some background on the company, and then we'll really talk about what we're looking to do over the next 12 to 18 months. The core technology of the company is a cell expansion system that we call bone marrow HPME, or HPME for high performance microenvironment. This technology was out-licensed from the University of Texas Health Science Center in San Antonio. The company was started about 4 years ago, and in its current form, with its current leadership, we've really been for the last 2 years or so. It is a platform technology. There are a number of interesting applications. I'll share some of those with you today. I suspect that some of you will, as you hear this and think about this, there may be some other ideas, and we'd love to talk to you about those as we go through this. From a strategic standpoint, we are focused on driving near-term revenues. We, to date, have focused on organic product development, but we are certainly looking at actively and considering a number of inorganic options. And again, if you have thoughts on that, we'd love to talk to you about it. Finally, from a strategic standpoint, we've done some early feasibility work on around a therapeutic application. I'll talk more about that in a second. And then finally, from a funding standpoint, we've completed an $8 million Series A round of financing just over a year and a half ago, and are about to launch a round of financing in the next few weeks here at Series B. I'm going to go through the next 3 slides pretty quickly. It's really to describe the core of our technology and what it is, and the best way for me to do that is to describe how we make it. So we start with commercially available bone marrow mesenchymal stem cells. We seed them into a dish or a other cell expansion format. We add media and growth factors, those cells expand and lay down a extracellular matrix. We then gently remove the cells at the conclusion of the production process, and what we have left is an extracellular matrix. And the reason we have found this so intriguing is as we've looked at data around what that matrix does, and here I'm sharing some data that came out of the Langer-Anderson labs at MIT, what we see in the first box on the upper left-hand side is you get more cells when you compare it to competing technology. The cells you get are smaller, and we would argue, therefore, more stem-like in their nature, potentially more potent. We've also done a fair amount of cell characterization on the cells that have come off the technology. And one of the markers we looked at and have looked at repeatedly is stage-specific embryonic antigen 4, SSEA4, and consistently across study after study, we see consistently higher levels of SSEA4 coming on the cells that come off our technology. The next question you might ask is, okay, you can do it once. Can you ensure that you do it across multiple batches? And we've spent probably the better part of the last 2 years getting the manufacturing of this right. The first thing we went out and did is with our partner BioBridge Global based in San Antonio, Texas, we built out a GMP-capable manufacturing space, about a 1,000 square foot suite, and we've been operating that now for about 18 months. We've also looked at donor consistency and across donor variability across lots, and as you can see in the chart there on the upper right-hand side, we see very consistent performance of the matrix across donors. In this particular one, we're looking at cell doubling time, and you can see on the bottom there, it's consistently better cell doubling time than in this case on tissue culture plastic. The other thing I'll mention very briefly is our patent portfolio. When we out-licensed the technology 4 years ago from the University of Texas system, we had 3 pending patents. Today we have 7 issued patents, both here in the United States and in countries around the world. We have a number of pending patents, including 2 that resulted from work we did internally that we will not be sharing with the University of Texas system. Those will be wholly owned by the company. So I've very quickly given you the foundation of the company, and when I became CEO 2 years ago, most of this was already in place. The real question for us was, so what do we do with this? And in working with our board and my management team over the last 2 years, we've really developed a three-pronged strategy, if you will. First, what we want to do is commercialize what we have into the research market. And it's both the HPME and, as I'll talk about in a minute, complementary tools around that HPME. And in fact, what we're looking at doing, we believe we have the foundation in place to begin to build out a life science tool company focused on regenerative medicine. Two, we believe that given that this is a platform technology, one of the best things we can do is find partners to help us identify the various uses that technology might have. And in fact, this is a strategy we've been pursuing very actively since this meeting 2 years ago. And we will have our first products we'll be announcing in the first quarter of next year that have come out of those collaborations. We are continuing to identify collaborators. We met with people today who may be potential collaborators for us going forward. We believe that this is a very, very important part of capturing the value of the technology that we have. And then finally, there is a clear hypothesis that we have, and Bob Langer, who sits on our board, has really driven this with our board, that the technology itself may have some very interesting therapeutic benefits. And to that end, we've been doing some early feasibility study to identify and confirm those benefits. What we have decided as a company is that for the foreseeable future, we will protect our very precious dilutive equity capital and won't deploy a lot of it here, but will rather be seeking outside funding to help us develop and identify further applications around a therapeutic application. So I want to talk first about the new products. I'm not making any product announcements today, but we're getting close. The first area where we see products, and again, we're going to make these, as we look now, these product announcements will all be in 2017. We've identified 6 new products that are in the works today and that we're committed to doing. The first, we have 2 cell lines that we believe are unique and proprietary that we'll be announcing in the first half of next year. In fact, I suspect one of those cell lines could be announced before the end of the year. Those came out of, as I said, collaborations that we have with other parties where we've taken their material, put it on our matrix, and actually enabled the expansion of cells that currently cannot be expanded via other means. The second is taking our current product, the HPME, for the research market and developing new formats of that. I suspect that the announcements in that place, in those cases, will be made in the second half of next year, and that's really been the focus of a lot of our internal R&D work. And then finally, the HPME, one of the clear questions we've always gotten is, what's the pathway to the clinic for the technology? And we made an announcement earlier this week that we were part of a $7.8 million grant that the military made that is with its end game to develop a clinical grade mesenchymal stem cell for use in particular with trauma victims in the military. Our matrix is part of that. We are committed to producing not only an animal-free version of the matrix, but also a version of the matrix that can be used for large-scale manufacturing. So there'll be a couple different products that come out of that work, and we are committed, as by the terms of the grant, to deliver that by the end of next year. The other thing we realized as we looked at the strategy earlier this year was there was a lot of potential in the research market, but it was difficult for a company of our scale and size to get. We did not have a budget to go travel across the country. There's only so many conferences you can do, and it really was going to be incumbent on us to find the right partners to help us get this product into the hands of researchers and to begin driving revenue for the company. So to that end, we identified a strategy of partnering with various distributors around the world. And over the course of the last 4 or 5 months, we've announced agreements in Japan with Funakoshi, South Korea with Solin, and in Europe with Cellaviva. Cellaviva is a Scandinavian company. They have exclusivity in Scandinavia, and then they have the rights to the rest of Europe. I had hoped against hope that I would be able to make a major announcement around a U.S. distributor at this meeting. The best laid plans of mice and men, as they say, that won't happen, but I can assure you and tell you that we are probably several weeks away, or maybe even several days away from being able to do that. The last thing they told me they needed was their name on our insurance liability agreement, and that happened yesterday, so we should be very close. In any event, we believe we will be in the catalog of a major U.S. distributor by the 1st of January. And we're very, very excited and believe that will be a real difference maker for us in this space. I talked about collaborations and what we've done. And again, we've had a guiding principle that the HPME technology is really at the core of all of our collaborations. And I've given you a flavor here of what those collaborations have done. And one of the areas we've gone after is where we've had partners who've had a cell line that is just very hard to grow. And as I say, in 2 cases, we've been able to take that, put it on our matrix, and in fact have been able to grow those cell lines into products that will be announced next year. We've taken the product in a different form and combined it with another company, other companies' products to enhance the performance of that product. And in one case, we've worked with a partner to grow cancer cells much faster and in much larger quantities than they were able to do with their product alone. And then finally, we've partnered with a couple groups to actually put this on a medical device and improve the performance of that device. Again, this kind of just gives you a flavor of what we've done. As I said, 2 product announcements coming out of these collaborations. And if you have an idea, we're certainly open to talking and working together on these. I'm going to very quickly talk about the therapeutic feasibility work that we've done. The chart here just looks at, we did an 8-week study where we took our matrix, we combined it with hydroxyapatite, and we looked at the impact on bone repair. This is looking at a snippet of time, weeks 4 through 8, and the fold increase. And Group B was the hydroxyapatite alone. Group C was the combined product. And you can see that we had a significantly higher level of performance of the combined product. While Group A, which was the control group, did better overall in the fold increase, I will tell you that the overall numbers were significantly lower on Group A. If you are interested in this, my colleague Cy Griffey will be poster number 1 on Friday, and Cy can give you all of the details on this study, what we found, and where we're planning to take this going forward. Finally, a comment about financing, because this is always interesting to people in this audience. As I mentioned, we had an $8 million round of financing that we concluded. I think some of our literature says April, some of it says March. The final deal was signed in March. We announced it in April. So if anybody's wondering, that's what it was. That was led by a $2.25 million investment from Targeted Technology, which is a San Antonio-based biotech fund. The city of San Antonio invested, and yes, for those of you who wonder, it really is easy and really great to do business in Texas, and I say that as a 25-year New Yorker. It's been remarkable. University of Texas invested in it, and then a small seed fund, T3DC, is also one of our investors, and the remainder were local angel investors, 50 or so. 48 of them were residents of San Antonio who were committed to regenerative medicine and biotech in San Antonio, and the other 2 were personal friends of mine. So there we go. Our Series B round we launched late last year. We raised $1.1 million, and then because of the final bullet on there, we actually suspended the financing because we entered in some discussions with a strategic partner who is about to make a pretty significant investment in the company, and we felt that it would be better off from a valuation standpoint to go out and finish out the Series B round once we were announced that. And again, this is something that will be very soon. I suspect it could be as early as early next week. Again, it's in the hands of the lawyers, and I think everybody's agreed to everything, but we'll see. No company is good without a team. We have a very strong management team, a great board. It's led by Kathy Burzig. Many of you may know Kathy. She's actually here in the room. Kathy ran KCI. She ran Applied Biosystems, and I consider myself blessed beyond all belief to have Kathy as our chairman. She's been a great friend and mentor to me as we've gone through this. A number of business people from the San Antonio community, as well as I mentioned earlier, Bob Langer, who brings a unique perspective from a scientific standpoint. That's all I have to say. We do have a booth here that we'd love to talk to you there. We will also have the poster session, as I said, poster number 1 on Friday, and we'd love to talk to you if you've got any ideas. Thank you.